Alright, welcome back to our last lesson of this unit and of course of Chem 30. So we're going to be looking at interpreting pH curves, so there's going to be a few different things in here. We're going to be looking at what we call polyprotic substances, so these are acids and bases that have more than one proton to either accept or donate. We're going to be looking at buffers, and then of course how that ties into our pH curves. So this is a little lengthy here, but everything kind of ties in together, so that's why I couldn't just separate it one lesson at a time. So first thing, let's look at our first little bit of definition stuff, and that is this polyprotic entities. So what we have is something called monoprotic. Just remember, mono means one, protic meaning proton. So a monoprotic acid is an acid that is only capable of donating one proton and one proton only. So therefore it can only react with water once to produce hydronium. So of course all of our monoprotic acids, you're going to notice they only have one acidic hydrogen on the left, or one COOH group. Now a polyprotic acid is going to be an acid that can donate more than one proton. So you're going to be looking at acids that have more than one hydrogen in their formula. So like more than one hydrogen on the left or more than one COOH group. So you can have that kind of group definition of polyprotic, or you can be a little more specific based on the actual acid. So if there's only two hydrogens, we call it diprotic. If there's three acids, we can call it triprotic. And that's as far as we actually go with our acids. So for example, H3PO4, so phosphoric acid, if this acid was be able to react 3 times, specifically with something like a strong base, it potentially can lose all 3 of its hydrogens. So for example, if we get our H3PO4 plus hydroxide, we can get H2PO4 minus plus H2O. Now, the reason I'm using a single arrow here is what we're doing is we're assuming that if we can get adding enough hydroxide, and this is why it's going to be a strong base, where we can get basically all of that first hydrogen transferred, so get 100% reaction. Therefore, there's going to be no H3PO4 left, but we're only left with the dihydrogen phosphate ion. So then in the second reaction, the dihydrogen phosphate can react with more hydroxide, and hopefully, once again, we're going to assume a quantitative reaction, so we can lose all of the hydrogen that's present. And then of course, if all of the dihydrogen phosphate is done, what we're left with our hydrogen phosphate, once again reacting with our strong base, assuming that quantitative reaction. And of course now we notice that there's nothing left. It's PO4 3 minus, there's no more hydrogens there. Oh, sorry, I just noticed the charges, I messed up. This one was right up here all along. I don't know why I tossed a 2 in there. And our HPO4 should be 2 minus. And that's, yeah, I had that right there. So that's kind of how we do these multiple ones right here. Now, of course, depending on what we have, you should be able to look at chemical formulas and determine if it's going to be monoprotic or diprotic. So for example, H2SO4, we know it is a strong acid, but we have our 2 hydrogens here. So it should be a diprotic acid. Now, I also said you had to look for that COOH group, that carboxylic acid functional group. So when you get something like oxalic acid right here, we see COOH off on the side here, COOH off on the side here. So we have 2 carboxylic acid functional groups. So this is also diprotic. Now for this one here, this is just methanoic acid. Remember, this hydrogen on the left, if you drew this out, organic chemistry wise, we'll notice that this is the only acid group right here. This one is not acidic because it's covalently bonded to that carbon, so this one's only monoprotic. And of course, that's just the first 3, so I just want you to pause the video and see if you can get the next 3, but the answers are going to go up in 3 seconds. And we get our answers that look something like this. Now, all the ones I have as diprotic, you can also list them as polyprotic. That's perfectly okay as well. And that's where it says label each as either monoprotic or polyprotic. So technically all the diprotic ones you can just label as polyprotic, and that's fine. Now, this whole thing about the, mono and diprotic, we can say the exact same thing with the bases. Right, so we know a monoprotic base is going to be a base that can only react with water or react once and only once to accept only one proton, whereas our definition of a polyprotic base is going to be a base that can react more than once. I know this is with water or an acid to form hydroxide ions, and it can accept more than one hydrogen. So of course, for depending on what we have, like showing the carbonate ion, how many reactions can the carbonate ion undergo? Now with this right here, remember that the negative charge on a polyatomic ion is going to attract the hydrogens. So this is going to be a diprotic base because that -2 charge. That means it can only react twice. Now I know I have up here in the definition react with water, but I'm going to have it react with hydronium in this case right here, because we're going to kind of assume the same thing. We want to react it with a strong acid so that we know it's going to completely take on all of that, or completely take the hydrogen ion that's there. So for example, if we have our reaction 1, our carbonate ion, it can react with an acid, now, same thing where we're adding the strong acid, we're assuming that we're adding it in excess so that it completely transfers the hydrogen onto the carbonate ion. So we have a quantitative reaction, meaning it goes to 100%, so it's not in equilibrium. We have all of hydrogen carbonate, and same thing where we have all of hydrogen carbonate react with an acid to produce our dihydrogen carbonate or our hydrogen carbonate molecule, which once we get to this, it no more negative charge, so it won't be able to attract another hydrogen. So of course the big thing with these here is, like I said, for the bases we look at the charge to figure out how many hydrogens it can take. For the acids we look for the COOH, or how many hydrogens are on the left. So just by looking at the chemical formulas, we have examples right here, so I want you to pause the video and see if you can look at our chemical species right here and write out the type of acid, if it's going to be a polyprotic or a monoprotic acid, and same thing with the base, right, if it's a monoprotic or a polyprotic base. All right, so pause the video because the answers are going to go up in 3 seconds. And when you get it done, it should look something like this. So for the acids, you just look at the left, see how many hydrogens are here. Now obviously for the water, it's not going to be diprotic because you're not going to get O2- as an ion, like it's either going to stop at hydroxide or hydronium. So water is always going to be monoprotic, but we tell here mono, mono, di, and mono. And for the bases, we look at the charge. So -1 charge, monoprotic, -1 charge, monoprotic, -2 charge, polyprotic or our diprotic, and then of course monoprotic and monoprotic. Now, since the polyprotic substances do have more than one proton to donate or accept, they can undergo more than one reaction if chemicals are continually added to it. So basically doing a titration to complete the reaction. And this is why for the bases I had the hydronium in here, showing that, okay, we're continually adding it. So we can predict what reactions would happen by using our 5-step method if we want to, because we have to remember only one proton is transferred at a time from the strongest acid to the strongest base. So basically what that means is when we complete one reaction here, like say for reaction 1, once the carbonate is completely used up, because we assume we get a quantitative reaction here, we produce nothing but HCO3- and then H2O. But because we're assuming that's quantitative reaction, once we're done, there's no more CO3- there's no more carbonate ion. So we completely use it up. And that's basically kind of when we're doing these kind of, I want to say titration type things, that we can predict how much is there. So we're going to assume that we have quantitative reactions in all of them because we're continuously adding stuff. So I'm just going to do example 1, just once again kind of showing you how that happens, and then I'll let you guys kind of pause the video and try example 2 and 3 on your own. So like for example with the reactions, potassium hydroxide is continually added to oxalic acid until no more reactions occurs. All right, so oxalic acid, if you want to, we know this is HOOCCOOH, and because we have this and this, so we have 2 carboxylic acid functional groups, we know it's diprotic. Now for our reactions, I'm just going to show you using the 5-step method. So for reaction 1, our entities, we have potassium hydroxide continuously added to oxalic acid solution. All right, so this is everything that's here. Now for that first reaction, obviously our strongest base is going to be hydroxide, and our strongest acid, when you check your periodic or your table in your data booklet, oxalic acid is going to be our strongest acid. So our first reaction is we get our base plus our oxalic acid. We get a transfer of 1 proton. Now, just like before, we're going to assume that it's quantitative because we don't want to have this in equilibrium. We want all of that hydrogens, or that first hydrogen, to be completely transferred from the oxalic acid. And our first reaction will look something like this using our 5-step method. Now, once this reaction's done, basically what we have is there's no more oxalic acid. We have our hydrogen oxalate. Because if we're continuously adding potassium hydroxide, so that means for reaction 2, we still have an abundance of potassium and hydroxide. But now we have this. We have our hydrogen oxalate ion. And of course, with this right here, because we're adding our potassium hydroxide, our hydroxide is going to be our strongest base. Now when we're looking at which one's going to be a stronger acid, our hydrogen oxalate or our water, from our data booklet, the hydrogen oxalate should be the stronger acid. So our second reaction, oops, that's not oxalate. So let me try that again. So our second reaction, we are going to involve. And we're going to assume that it's quantitative again because we want to completely remove that last hydrogen. And that is it. So of course, if all of our hydrogen oxalate ions gone, we only have our oxalate ion, so OOCCOO2 minus. We'll notice that when we write everything out, there's no more acids present besides the water, but if you get a transfer from hydroxide, a hydrogen from water to hydroxide, we're going to form water and produce hydroxide, so there's no reaction left. So that's how we use our 5-step methods to actually predict, showing how each reaction occurs. But like we done in the past there, or just on the previous slide, as long as you can realize if it's supposed to be monoprotic or diprotic, you can kind of jump the gun and do kind of what I did here, and realizing that we know the carbonate ion is diprotic, so it can react twice with hydronium to produce the subsequent conjugate bases or conjugate acids. So that means I want you guys to try, or pause the video and try example 2 and 3 on your own, and I will have the reactions here now in a second. So once again, pause the video, 3 seconds, the answer's going up. And your answers should look something like this. So our sodium hydrogen phosphate, our hydrogen phosphate is a diprotic base, so we have 2 reactions, and of course the net, when we add them together, we'll notice our dihydrogen phosphate could cancel out, and we're just left with this as our net reaction. And then for example 3, we have the borate ion, so BO3 3-, that 3- indicating that it is triprotic, and we get our subsequent reactions right here. So triprotic, we have 1, 2, 3 reactions. Now you will notice that we're reacting with H3O+ because we have hydroiodic acid, for example 2, hydrochloric acid in example 3. These are strong acids, and remember with strong acids, when we write them out in entities, it's H3O+. Now, the whole reason I kind of wanted to introduce this whole monoprotic and diprotic bases and acids, specifically those weak ones, this kind of ties in a little bit with something called buffers. Now, you've definitely learned this word before if you've done biology and stuff like this. You would have learned that buffers are kind of everywhere, and what they do is just resist pH change. And that is actually all they do. So buffers just have a unique ability of maintaining nearly a constant pH, and this is all they're going to do. So we're going to look at how they actually occur, or actually how it occurs, sorry. But this is the main thing that you need to know with buffers, is they maintain a nearly constant pH. So basically for us, what that means is if you add an acid or base into a buffer system, the pH will not change at all. So how do we make a buffer? Well, a buffer needs to have a weak acid and a salt of that acid. And that salt is the conjugate base of the acid. So we call it a salt because if you look at some of our weak acids, like if you want to make one out of hydrofluoric acid, we need HF, but we need the fluoride ion as well, right? The conjugate base of the weak acid. Now the thing is, you cannot pick up a jar of fluoride ions. It doesn't exist. You can pick up something like sodium fluoride, lithium fluoride, so a soluble ionic compound that can get that in there. Now for soluble ionic compounds, chemists generally call them salts. So it's not just sodium chloride, it's an ionic compound, a soluble ionic compound of the conjugate base. So that's referred to as a salt. And that's why I have right here, a salt is a common term used for a soluble ionic compound. So, example of let's say a buffer system. Made from, let's see, the carbonate ion, or sorry, that's not carbonate, acetate ion. We can tell we can create a buffer if we have acetic acid and sodium acetate. So we need that sodium in here because when we dissolve it in water, this is going to break apart into sodium and the acetate, but we need to have the weak acid, and this right here, just the acetate, is a conjugate base of that weak acid. Now, of course, buffers are going to be used to calibrate pH meters if you're doing very sensitive reactions. And then, of course, for us, it does help us control pH sensitive reactions in the blood. Now, just to kind of show you on how the buffers actually work, so why they can protect against either an acid or a base getting added to something. So I'm just going to show you how an acetic acid sodium acetate buffer maintains a constant pH when small amounts of hydrochloric acid or small amounts of sodium hydroxide added. So if we add HCl, add NaOH. Alright, so if we add HCl, well, the entities in our system, if we have our acetic acid sodium acetate buffer, so we have our acetic acid, so that's CH3COOH. We have our sodium acetate. And then, of course, if we add our HCl, that's adding H3O plus and Cl minus. Now, from here, you're going to go to your data booklet. You are going to find that our H3O plus is our strong acid, and the strongest base, I forgot to add water as well, sorry about that. That's also an entity, but that won't react because our strongest base is going to be our acetate right here. So what happens is that our conjugate base in our buffer, our sodium acetate or our acetate, can actually take the hydronium ion from our hydrochloric acid and it can neutralize it. We'll notice that we're going to produce more acetic acid, which we already have it right here anyway, and we know weak acids ionize to a very, very, very small degree, so producing a little more of a weak acid is not going to affect the pH at all, because they only ionize at like 1% anyway, so we're actually not affecting the pH whatsoever, but we are completely getting rid of our H3O+, and of course we know products are favored for this reaction. So we're essentially going to be using up all of our H3O+. Now, of course, for our addition of NaOH, same thing is that when we write out all of our entities, we got our acetic acid, we got our sodium, we got our acetate. Now this time for our sodium hydroxide, we're adding sodium again. Actually, I'm not going to write out sodium again because I don't need to, because we already have sodium over here, but we know metal ions don't do anything. So hydroxide and water. Now for this one, when you go to your data booklet and find out, hydroxide obviously going to be the strongest base, but in this case right here now, our strongest acid is our acetic acid. So now, by adding small amounts of our sodium hydroxide into our buffer system, now our acetic acid can step in and it can react with the hydroxide to produce our acetate ion and water. And same thing, products are favored. So we're going to produce most of the acetate ion here, which is already in solution. It is being a weak base, so the weak base being produced will not affect the pH whatsoever. But the main thing is that the hydroxide that's getting added is getting neutralized. So that is why these buffers will work, and they help protect against the addition of either an acid or a base. So just to kind of summarize, the acid in the conjugate acid-base pair of the buffer protects against any base that's added, and the base of the conjugate acid-base pair protects against any acid added. Now, buffers can be overwhelmed if too much acid or base are used, but the thing is you have to remember is that they maintain a constant pH, and that is the main thing right there. So we do have just some buffers right here that are kind of important. So in our living cells, I believe we have the HPO4 minus and our H2PO4 minus buffer. So this is actually the base listed first, but we have our weak acid conjugate base. So just let me adjust that just so not to confuse anyone. Nope, sorry. And in our blood, we have our HCO3 minus and our CO3 2 minus. So these are kind of important buffers inside of our blood and our living cells. Now, of course, next we do have a few little old diploma example questions. So I just want you to read this through, pause the video, and see if you can get the answer, because the answer is going to go up in 3 seconds. And hopefully you got A. So for this particular one, we actually have the carbonate ion react with hydronium to produce that. And then, of course, the whole thing, when we look at what's given right here, we have our calcium carbonate. That is why the buffer is going to be this. And what is the effect of the buffer? Well, it maintains a constant pH always. So you could have easily either taken out B and D based on the fact that a buffer will not change a pH. And what we have right here, once again, this one is a little more challenging to kind of figure out, but just by looking at it really quick, just read the question before we actually look at all the information up here. So which of the following rows identifies the observed color and the observed effect on pH after addition of small amounts of HCl to each of the solutions? So what we have right here is we got an H2S solution. So of course no buffer, and we have a buffer system right here. and they're starting with the same color because of the indicator, and they're starting with the exact same pH. So if we add a strong acid to our first solution that has no buffer, the color should change and the pH should also drop. So just on looking at our effects for solution 1, knowing that it's not a buffer, it's only a weak acid, so then of course if we had strong acids, we're going to decrease the pH. So just by looking at our effects that could happen, solution 1, no change, decrease. Oh, it has to be C, right? Just because I know by adding our acid that's going to happen, but then just also to make sure, so we have our buffer solution right here, so the pH should not change whatsoever, and of course no change. Now as for the colors in your data booklet, you can look at the indicator colors here and try to figure out the color stuff. So that is a little bit on buffers. Now, our last thing is just going to be looking at tying two of these things together, so our polyprotic and our monoprotic acids and bases and our buffer systems, and look at how they tie together in a graph. So just remember titrations. Titrations can be used to determine the pH or endpoint of an acid-base reaction, or even we've done them before with just trying to find concentrations of stuff. Remember, concentration of hydronium and hydroxide relate to pH. So an information from a titration can be plotted on a graph. We can look for buffer regions, can be analyzed, stoichiometric calculations can be formed, and pH of sample and titrant can be found as well. So that is just a bunch of information that we can pull from acid-base titration graphs. Now, these are called pH curves. No reason because the graph's going to curve, but is just a graph showing the continuous change in pH during an acid-base reaction. Now, for these right here, the pH is going to be on the y-axis, and the volume of titrant added is going to be on the x-axis. Now, for us, we're going to tell when an endpoint is going to happen. This is going to be the endpoint of the reaction when it's gone to completion. Usually, it's by a color. Oops, almost spelled color wrong. Normally, we do recognize these by changing color. And another thing for our graph is going to be expressed as the vertical region on the graph. So that's how we know our endpoint is going to be reached. And of course, equivalence point is just our stoichiometric amounts of reactants added are equal. So number of moles of our given is equal to the number of moles of our wanted. Now, a new thing that we're going to be looking at, a buffer region. So we already know the endpoint is the vertical region. So remember, vertical is up and down. So if we see a sharp change right here, that is going to be the endpoint. Now the buffer region is going to be a horizontal region. So basically a flat region at the beginning of a pH curve, when the pH of the sample remains relatively constant with the addition of our small amounts of acid and bases. So of course we're going to look at graphs now in a minute and kind of tie all this together. But as we're interpreting these pH curves, just remember the initial pH of the curve must equal the pH of our unknown solution, right, or our sample. And the endpoint and the equivalence points are usually located in the middle of the graph and the steepest point, steepest drop on the plotted line. And after excess titrant has been added, that is going to be the pH near the titrant. So that's how we can figure out all of these different things. So, and also we're going to see multiple equivalence points or multiple endpoints depending on if it's monoprotic or diprotic. So the number of equivalence points must match the number of quantitative reactions. Now, of course, we are going to be looking at graphs very soon and showing all this, but last but not least, we're just going to look at selecting appropriate titrants. Now, to select the correct titrant to use in a reaction, first of all, we need to determine if the sample is an acid or base. So if the sample is an acid, Whether it's strong or weak, the titrant should be a strong base such as sodium hydroxide or potassium hydroxide. This is just to make sure that if we are doing our reaction, we need to have at least one strong entity in there to try to get a quantitative reaction. Whereas if the sample is a base, we're going to use, the titrant should be a strong acid, specifically monoprotic, to minimize second reactions. So out of our 6 strong acids, basically 5 of them should be there. So monoprotic, 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 and monoprotic. Sulfuric acid we are not going to use for titrations. Just because it is diprotic, because we can get secondary reactions based on the hydrogen sulfate ion that's there. So we tend to leave out sulfuric acid just because. Now also, if you do read the word neutralize, it doesn't mean that it's going to bring it directly to a pH of 7. It's just meaning it's bringing the pH closer to 7, right? So it's either we take away the acidic properties of an acid or we take away the basic properties of a base and bring it closer to neutral. So let's look at our different curves. So generally, this can be what we would expect for a strong monoprotic acid reacting with a strong monoprotic base. So you'll notice the endpoint is right in the middle, right at a pH of 7. Now, generally we can tell as well, so at the beginning, we can get the pH of the sample. So at the beginning, before any titrant is added, that's going to be the pH of our sample inside of our Erlenmeyer flask. Whereas at the end, once the sample is used up and we're just adding in excess titrant, we get the pH of our titrant at the end of the graph. So for our first graph right here, we can tell our sample is acidic, our titrant is basic, the titrant almost has a pH of 13, our acid at the beginning almost has a pH of 1. Then of course, same thing here, we can tell that we have pH of our sample, pH of our titrant, and our equivalence point at a pH of 7. Now for our strong monoprotic and strong monoprotic, both acids and base, there's no buffer regions. And the reason for that, a buffer needs to be a weak acid or base with its conjugate acid or base. So where we're dealing with a strong monoprotic acid and a strong monoprotic base, there's no weak one, so therefore no buffer. Now, for our weak monoprotic acids and our weak monoprotic bases, you'll notice that the curves look a little different. So we can still pick out the exact same thing. So we get our pH of our sample at the beginning. We get a pH of the titrant at the end. But notice what we have right here. We can see we get a sharp decrease in pH, but then it starts to level off right here. Now I know it's not completely flat, but this leveling off portion right here, this is our buffer region. And the reason it is a buffer region because we can tell it's not following the trend here. We get a sharp decrease, then the buffer starts to kick in, so it starts resisting the pH, and then it's overwhelmed at some point. Now, because we're starting with obviously a weak acid up here because the pH is much lower, we get the equivalence point to be much lower as well. So that's why if we start off with a weak acid, the pH of the equivalence point should be less than 7. Whereas a weak base, the pH is going to be greater than 7. So these need to be changed around here. And we can tell the exact same thing right here. We see our buffer region in the equivalence point. Same thing because we're starting with a weak acid. So the pH, oh sorry, these were actually correct. I was looking at the wrong graph there. Sorry, didn't mean to confuse you guys. So if we are starting with a weak acid, we're using a strong base, the pH is going to be above. Yeah, I was looking at this graph right here for that one, and sorry, my bad. So these are correct in saying that. So if our sample is a weak base, the equivalence point is going to be a little lower. If our sample is a weak acid, the equivalence region is going to be a little higher. And the reason for this is that our titrant is a strong base if we're using a weak acid as our sample. So if it's a strong base, therefore the pH is going to be closer to that. Now, last but not least, just looking at some of these curves, if we have a polyprotic substance, depending on what we have right here, we can get different regions. So this is where we have, we can tell we have 1, 2 equivalence points. So this is a diprotic acid for our sample, right? Because it's starting weak right here. So same thing, we see buffer region 1, then the buffer is overwhelmed, first equivalence point, and then the second buffer kicks in right here, flattens it out, it's overwhelmed, boom, our second equivalence point, and then finally, same thing, we can get our pH of our titrant, and at the beginning, we get a pH of our sample. Now, this is just kind of a summary right here, so I'll let you guys kind of read that. So our sample is always in the flask, titrant's always in the burette. So this just kind of tells you initially pH, final pH, and basically where the equivalence point is. But you don't really need to worry too much about these general things here, because if you're asked to solve any questions about it, you're always given the curve, and it's just going to be where's the buffer regions and stuff like that. So, last but not least, choosing an indicator for these reactions. So what we have is, or what we need is that if we're looking at an indicator, and you guys done this with acid-base reactions before, it's basically one drop during our titration and we get a permanent color change. So an appropriate indicator will completely change color within the steepest portion of our pH curve. And the reason for that is, if you can imagine, if you had an indicator that changed basically from these pH, like notice how much volume you need to add, whereas if we get an indicator that changes basically right around 6, there's very little volume of titrant added here, so we're going to get a very sharp change in color over a very small amount of titrant added. So that's what we're looking for. So all we need to do is, well, look at our indicators. Oh, sorry, not these right here. Indicators in your data booklet, which we have their pH range right here. Now, all you need to do is make sure this pH range, so if we're using something like phenol red or methyl red, is that this pH range is going to fit or change within our steepest portion. So when we look at, the steepest portion right here, let's just say this first equivalence point, right? So steepest portion takes in basically about that pH of 6. So if we want to choose an appropriate indicator for that particular reaction, I would probably use chlorophenol red, because that pH of 6, so the equivalence point was 6, that pH of 6 is right in between our 2 points right here. So that means that it would probably change color during that steepest portion. Now, with some of these right here, there's always going to be multiple answers, but that's okay. And that's why I have right here, so we have different indicators. Now, this is out of Chem 20 as well. If it's in the lower region of the pH, it's HIN. If it's in the basic portion, it's IN minus. Now, these are just an example of some of the ones right here, how we get abbreviations in their pH range. So litmus, we know when it's in the acidic form, it's red. In the basic form, it is blue. Now, every acid and base is going to be different. Now, to show the equivalence point of an acid-base titration, we choose an indicator whose color change range includes the equivalence point, which I just showed you. So that is how, if you look at any graph, as long as the pH range is in right there, and now even with the previous one on the previous page, even bromothymol blue might be good because this range is close to, or even the litmus might work as well. All right, now last thing, just kind of bringing into the whole thing with the polyprotic and the monoprotic ones right here, is that if we do have any samples are polyprotic acids and bases. Of course, we do know those are substances that can donate or accept more than one proton. Now, as we start taking away or giving up or accepting more hydrogens, it becomes more difficult to remove the second or third protons, because as we start donating more protons or accepting more, everything becomes weaker. So normally the removal of the first proton or the donation of the first proton in any polyprotic substances is generally quantitative, whereas the second and third ones are generally non-quantitative unless otherwise stated. So only quantitative reactions can produce detectable endpoints, because those are the only ones that have the steep portions. So, last little diagram we're going to look at, then you guys can answer the last question. But this right here, just kind of bringing into that, we have a titration of H3PO4 with sodium hydroxide. It shows 2 endpoints. Now, even though we know this is triprotic, there should be 3 endpoints, but it's saying that there's only 2 endpoints because, well, the removal of that last hydrogen is almost impossible. So that means if you are to draw the pH curve, basically we draw 2 endpoints, right? So you start here, we get a buffer region 1, we get M.1, then second buffer region, M.2, and then that's it. And that is just taking this whole idea of we're only showing 2 endpoints. So this is what it would look like. Now, just before, or just as before, you would need to kind of have this right here to kind of let you know that 2 endpoints are actually drawn. But once again, you guys won't need to worry about drawing them too much. For the most part, yours is going to look like this, where you're given a titration curve. We can tell that there's 1 endpoint here, 1 endpoint right here. So we have a polyprotic substance. And when we're answering the question, so in the titration represented by the graph above, the titrant that is added to solution is blank, and the sample is blank. Now, the fact that I just pointed out 2 endpoints, we know it's polyprotic, so we can not worry about A, not worry about C. So our answer is either B or D. Now, when we look at it, remember this is the pH of our sample, and our sample is a base. This is the pH of our titrant. And when we looked at here, pH is below 7, so we can tell it is an acid. So that means our answer is D. And that is it for Chem 30. So this week I am going to post the assignment that was for last week, and then of course the assignment, the unit assignment, that'll go with this entire unit as well. So you guys try to finish that, with the time that you have. If you're not sure of anything that was kind of covered over, I know this week the lessons are a lot longer. That's because there's so much that's in these right here, and they all kind of feed off each other. So I couldn't really chunk them into smaller sections, because then it just wouldn't flow as nicely, or they wouldn't really go into each other as nice there. So we kind of had to do it this way, which I apologize for the longer lessons. But anyway, make sure you continue your work. Make sure you're still working on your workbook. If you have any questions, make sure to email me. And of course, hopefully I'll be able to see you guys at some point for your graduation, or who knows, just whenever. Hopefully all this stuff will clear off. I'll be able to see you guys soon. Anyway, have a great summer. Show up for the online little help sessions, and I'll talk to you guys later.